So, we begin with the second part of the course from this lecture onwards. In this part we will look at how protein structures are solved by NMR, what kind of isotope labeling techniques are required, how 3D NMR experiments are used for assignments, and in the last part of the course we will look at how protein ligand interactions can be studied. So, let us begin with a very basic understanding of what is a protein structure, how does it look, what are the different aspects of a structure of proteins, so that we can understand how NMR then can be used for solving them. So, this is what is now we start from. So, we looked at from the very beginning how the nucleus is made up of nucleons, and that is how the NMR began, looking at the spin of the nucleus, and that is what is the most basic element in an atom. Of course, this can further be broken down, but we will look not go beyond this nucleons. So, given this is the most basic level of organization, that is most basic elements from which all nuclei in this universe are built. Now, these nucleons are packed in the nucleus, these are very tightly packed, and there are a lot of great force which holds them together, and nucleus is located in an atom. So, atoms are made up of nucleus and electrons which are circulating around the nucleus, and that is the concept of atoms. And atoms actually organize or join together to form molecules. And this is something which all of us know, but what is important is beyond molecules. So, if you look at molecules which are made up of atoms, molecules themselves can arrange or self assemble or organize in different hierarchical structures. They can go into different complicated complex structures either by self driven or it can be through induced assembly. So, these higher order structures which are formed from the molecules are known as macromolecules. So, macromolecules is a word basically for molecules such as nucleic acids, DNA and proteins or membranes or lipids bilayers. So, these are complex molecules. So, we will look at how proteins can be studied by NMR, because the other two are very complicated on their own, they require altogether different topics to be covered. So, we will look at protein, but down the line we can also look at how protein interact with these different components such as nucleic acid and membranes. So, this is just a schematic of how the different levels of organization in a living system. So, we are looking at atoms, actually nuclei by NMR, which are located in the atoms of these big molecules. So, we will be probing by NMR every nucleus here in this molecule. So, by probing every nucleus in this molecule, NMR is a very unique technique which allows us to get a very high resolution information. High resolution meaning we can get atomic level details of each and every atom in this molecule, because each and every atom will have a nucleus and that nucleus can be studied by NMR. So, that is a unique aspect of NMR, that it gives us high resolution information or structural details into molecules or big molecules. So, this is the central dogma in biology, which again is very well known, that at a very basic level you have a genome, that is DNA, which is there in the nucleus, and that is transcribed into RNA, that is it is transcribed into RNA, and there are different types of RNA, but finally this RNA, messenger RNA, which codes for a protein, is translated or converted into a protein in the cell, and that protein then which is produced carries out the different biological functions. So, our focus is going to be on this aspect, how protein carry out the different biological functions. So, this field where we study the structure of a protein or its interaction with a gene, or how does a gene function or a protein function with respect to its structure at a molecular level, that means at an atomic level, that field we call it as structural biology. So, structural biology is a very emerging area where we look at different aspects of how a protein structure correlates with its function and how does it interact with different ligands in the system. So, these are some basic aspects what we are covering now. As we go down the line we will look at some more details. So, for example, now some proteins, not all of them, again we will look at proteins which are different from this. So, some proteins in universe, in our body, in a cell or in an organism, they have a specific 3-dimensional structure, and this is very important to bear in mind, because proteins are actually not just simple molecules, planar molecules, they are 3-dimensional molecules, complicated or macromolecules, where we have to understand how that structure is formed, how it works in space. So, this is a schematic again of some protein, which is shown in an animated mode. So, you can see here the red color turns which are seen here are the helices, alpha helix, and the blue color, light blue cyan color things are the beta strands or beta sheets, and the gray color here are the loops, and you can see several side chains of amino acids shown in yellow. This is the stick model of amino acids. So, for example, this you can see here is an aromatic amino acid, hydrophobic, hydrophilic, and so on. So, one of the special feature of a protein, that is a globular protein which occurs inside the cell or outside the cell, is that the hydrophobic groups are located inside the protein structure. So, we can see inside this, that it is buried in the core of the protein, are the hydrophobic amino acid such as leucine, isoleucine, methionine, phenylalanine, alanine, and so on. Whereas, on the surface you will have more hydrophilic amino acids, like this blue color here is lysine, glutamine, aspartic acid, asparagine, and so on. So, that is the interesting the part in a protein, that the inside is hydrophobic, outside is hydrophilic, but only happens that can happen only in a globular, when it is inside a cytosol or outside the cell, not in the membrane case. So, membrane protein are actually opposite. There the hydrophilic groups are located outside the surface, on the surface, whereas the hydrophilic are inside. So, the hydrophobic is outside, hydrophilic is inside, is exactly opposite of what you see in a globular cytosolic protein. And why is that? Because the membrane environment which we saw in this slide here, let me go back, you can see here the membrane is actually hydrophobic in nature. So, the amino acids which are facing this bilayer, they should actually be hydrophobic, because hydrophilic cannot be attracted to hydrophobic groups. So, hydrophobic interactions make the hydrophobic part of the protein interact with this, that is outside of the protein, whereas inside the protein hydrophilic groups are then located. Now, this gray color, you see there is again a slight difference in the thickness. This is very thick here, then this thins out. What does that indicate? That is a schematic again to indicate the dynamics of the protein. So, what you see here on the screen, we are only seeing a rotating protein structure, we are not seeing how the different dynamics groups are dynamic with respect to each other, because the protein actually breathes, it fluctuates, all the bonds are moving and fluctuating at different time scales. That kind of a dynamics is not captured in this picture, but what is being captured is that how the loops are having the higher dynamics. So, the thickness here indicates that the loops are having more dynamics, means this loop, for example, here can span a wider range of conformation, and therefore, it is thicker compared to these helices here, which do not span a large range of conformation, they are rigid. So, when they are rigid, their dynamics is less, but loops which are between the helices and beta strands, they are always very dynamic, because they do not have any particular structure. So, this is just a general again introduction to protein structure, which you can refer to in many of the books which was shown in the beginning, and let us continue with this, look at some more basic aspects of protein structure. So, now this protein is made up of a linear sequence of amino acid. So, you can see this is a code, letter code, single letter code for alanine, G is for glycine, R is for arginine. So, each of the 20 amino acids have a single letter code assigned to them. So, for example, Y is for tyrosine. Okay. So, there is not direct correlation with the first letter, because tyrosine has T, but T is something else, this is threonine. So, I would recommend very strongly to actually memorize that what is the letter code stands for, because when we go down the line, when we look at protein assignments, we will not be again referring to the, we will be referring mainly to the single letter codes. So, if I say immediately RTY, one should be able to recollect that this stands for arginine, threonine, tyrosine. So, they are each amino acid as therefore a single letter code. So, now you can look at this sequence of a protein. This is called the primary sequence or primary structure of a protein. It consists of a linear sequence of amino acid, linear, there is no branching here. This could be the number of amino acid, depends on the size of the protein. For example, if it is 100 amino acid protein, it will be about 100 into 100, 10,000 molecular weight. So, the molecular weight will become around 10 kilodaltons. So, this is based on the fact that an average amino acid, a weight of average molecular mass of an amino acid is 100 or 110 daltons. So, now this linear sequence of amino acids, if it is allowed to fold, it starts folding into a 3-dimensional structure, but it first actually undergoes also a folding to secondary structure, which is not shown here. The secondary structure is basically alpha helix, which is shown here, beta sheet. So, they start forming first, and then these alpha helix and beta sheet come together, and then they form the final 3-dimensional structure. So, this is the concept of protein folding, okay, and this happens very fast. This from sequence, as soon as the protein is coming out from the ribosome, it starts folding like this. So, it is happens at the scales of milliseconds, sometimes very fast can be microseconds, but typical millisecond is the time scale for protein folding. So, that is very fast from NMR point of view. So, NMR actually what we do is we do not study this process of folding very often in a real time. We study the final 3-dimensional structure which is already formed. We can study the intermediate structures from here to here. When you go, you can study some kind of intermediate structure if you can arrest or you can hold that structure at different time points. That is something which we will not do in this, look into in this course. There is a large area of research where people have tried to track how the sequence of amino acids, a linear sequence, gets folded into a 3-dimensional well defined structure. Okay. So, this is the protein folding problem, but what we do in NMR is we will look at the final structure. That means, we want to solve the structure of this part, I mean we want to see how this looks. We will have this information with us, and using different NMR experiments we will have, our goal is to get this structure. So, that is our goal in this whole concept of protein structure determination. Now, proteins can actually be divided into different categories. As I said earlier, one category we can say globular, that is intracellular or extracellular protein. This means it is inside the cell or it is outside the cell, and once proteins which are sitting in between, that is on the membranes, they are called as membrane proteins. So, there is a fundamental difference between the two. As I said here, the hydrophobic groups of amino acids are inside the core of the protein, and the surface of the protein is made up of hydrophilic amino acids, whereas here it is exactly opposite. You have hydrophobic amino acid outside and hydrophilic amino acid are inside the core of the protein. Now, these proteins, both of them, can be further categorized into 3 categories. Number 1, they can exist as a single domain protein. A single domain is an example I just gave in the previous slide, where the entire chain folds into a single structure. Okay, so, this is called as an independent folding unit. So, if you take the sequence which of this protein, it will form as 3-dimensional specific structure, but it will remain alone, means it will not associate with any other structure. It will be a single chain structure. But you can also have what is called multi-domain proteins. So, if you look at this here example, the multi-domain means this is one protein, you can say independent, this is another independent protein, and they are actually joined, but there is a single chain again, meaning it starts from one end of this protein, let us say here, and goes all the way up to the end of this protein which is somewhere here. So, this is a single chain, means it is a single chain, but it has got divided into 2 parts, and these 2 parts are linked by a linker. So, this is a linker, this loop here which you can see in grey color is a flexible linker, meaning it is flexible. So, this domain and this domain are actually independently moving. they will not move together, this will have a separate movement, this will have a separate movement. But they are actually also independently folding, meaning if I take this only this part, cut it out here, suppose I cut out this protein here and I take only this part of the protein, it will get the same structure independent of whether this was present or not. Similarly, if I cut out this here again and take this portion, put it in water, it will get this, now adopt this structure which is independent, means it is does not depend on whether this part was available or there in the protein or not. So, therefore, they are kind of independent folding units. Then you can have another type of structure where different units, that is this, for example, take this single domain protein, multiple units of single domain protein, they associate with each other and form what is called a multimeric protein. So, multimeric proteins are those which have multimers, trimer, tetramer, dimer and so on. So, you can see this is a example of the famous hemoglobin molecule which is a tetramer in the functional form. Okay. So, this is how many proteins work, they do not have a single unit, they have either multi domain or 2 or 3 or more, and or they can actually even associate with more of the. So, here you can see they are not single chain, this red and blue are actually independent, they are separate molecules, they can be easily broken into separate 4 separate molecules using some treatment. So, they are not covalently linked. So, if you look at this picture again here, there is a covalent link between the protein, this domain and this domain, they are not loose separated, but here they are not covalently linked, they are non-covalently associated because of some sort of interactions, and therefore, they can be dissociated also. Okay. So, these are 3 different categories, and we will see that for different types of proteins we need different types of strategies in NMR to solve the structure. Now, further each of these now can further be associated with metal ions. For example, you can see here I have shown in grey color, these are metal ions. Now, these metal ions are because of the requirement for the protein to function. It may be a protein which needs a metal ion for function for catalysis, an enzyme, or it may be that it is storing those metal ions. In this case, this is example of a calcium binding protein. So, this grey color balls here are actually calcium ions. So, this protein is known to store calcium when the calcium becomes excess in the cell, but this is one type of metal ion. You can also have different types, and these metal ions can be again categorized into diamagnetic and paramagnetic. Paramagnetic metal ions are those which have an unpaired electrons, and that has a lot of implications in NMR, because paramagnetic causes ions, metal ions, they cause very high relaxation, very fast relaxation of protons which are surrounding this. For example, let us say assume that this is a metal ion which is paramagnetic, all the protons which are in this zone here will start getting broadened. Broadened means the signal will start disappearing because of this interaction of paramagnetic dipole moment with the nuclear dipole moment, electron and nuclear interaction. So, that becomes very challenging for NMR to be used, because broadening of lines means the sensitivity goes down. So, therefore, paramagnetic metal ion protein structure are very challenging part and has been dealt with by several groups, and that is again we will not go specifically into how to solve paramagnetic protein structure in this course, but you should remember that these metal ions are also part of the protein, they are required for the function of the protein. So, when we solve a structure, we should be aware whether a metal ion is there in the protein or not. There are varieties of techniques to find that out. You can refer to any spectroscopic method, and that can be used. Now, you could also have cofactors. For example, look at this here, hemoglobin as a heme, the porphyrin ring is a cofactor. So, these are also required because hemoglobin's main function is for transporting oxygen. So, oxygen has to bind, and there is an iron here. So, that is metal ion, again paramagnetic, and this porphyrin ring is holding that metal ion, and it has very important role to play, and that is that is called a cofactor. Now, the other thing is ligand. So, sometimes the protein interacts with DNA, or protein can interact with another protein, and then only it will function. So, again that always in a cell it may not exist like this alone, it may exist in a form in which it is bound to the ligand. The ligand can be any small molecule, metabolite, or it can be a small part of DNA, RNA, etc., or a peptide, so on. So, ligands are also part of the protein in the sense that they are required for the protein to work. So, the native protein always exists bound to the ligand in the cell. So, these are the other next level of complications, that is based on the each protein can further have these things available or present in its structure. So, these are again the basics of protein structures. Now, let us see what happens if the protein structure goes defunct. So, for example, we know this is a very broad picture that a biological function, that is what kind of function it has in a cell, depends very much on its 3D structure. So, for a proteins which have 3-dimensional structure. So, if I disrupt the 3D structure of the protein for some reason, for example, it can be mutagenesis, amino acid in a protein can get mutated to some other amino acid, or a range of a protein can amino acids can get mutated, or the protein may get denatured because of some acidic or alkaline conditions. In such cases, the 3D structure is lost. So, when the 3D structure is lost, whatever function it was carrying out now cannot be carried out. So, that is called as loss in activity, and this results in various problems in the cell or in the overall organism. For example, if you have Alzheimer's disease, there you have a protein which does not properly form the 3D structure, it gets misfolded and starts forming amyloid fibrils, and that fibrils can lead to plaques in the brain which cause the problem. Similarly, many other proteins, sickle cell anemia is a well known disease, a well known problem in which one amino acid in hemoglobin is mutated, and that results in a change in the structure of the protein and loss in or reduction in activity. So, like that there are many possible changes in structure happening in our body which can result in the structure getting disrupted or changed or basically damaged, and that can result in the malfunctioning. So, therefore, the goal of NMR and drug discovery and so on is to find out what makes the 3D structure go bad, or how we can retain the 3D structure by interacting with some ligand. Sometimes some ligand, if you add to the protein structure, it gets stabilized. So, can we study that kind of interaction and stabilize the protein structure. So, 3D structure is therefore very important to study for proteins, because their function or the actual role in our cell depends on that structure. So, how do we determine 3D structures of proteins? There are two main categories. One is called experimental method, or you can use completely theoretical approach. So, in experimental approaches, the well known methods are X-ray crystallography, which is a very old technique, and this is now well established, and a lot of protein structures are now being solved with X-ray. And second approach is what we are going to look at, that is NMR spectroscopy. In one difference between X-ray and NMR, an important difference is in X-ray you need the crystals of the protein. So, many proteins do not crystallize, therefore, those kind of proteins you have to do with NMR spectroscopy only, you cannot use X-ray. But the disadvantage of NMR is the size. Typically, as a protein goes beyond 50 kilodaltons, it becomes very difficult to solve the structure easily. In fact, up to 40 to 50 also is a higher range for NMR spectroscopy to get high resolution structure. So, remember, our goal here is high resolution, meaning we want every atom or every amino acid in the protein to be resolvable or separable, or we should know the coordinates of all of them. It is not just we want to know whether it is having alpha helix, beta sheet and so on, we want to actually know the 3-dimensional structure. So, for NMR the limitation comes from size. Higher the size of the protein, that is beyond 50 kD, it is very difficult even with modern techniques. So, crystallography does not have that limitation, but for crystallography you have to crystallize, and many times many proteins do not crystallize, and therefore, that is the difference advantages. They are basically complementary methods. We will see shortly that there is another class of proteins which again cannot be solved with crystallography, only NMR can help us there, and we will look at that in a few slides from now. Now, experimental technique another which has come up recently as becoming popular is cryo-electron microscopy. Again this requires the freezing of the molecule, like here freezing meaning crystallization or cryo, which is frozen. So, again this is looking at a static structure. So, that is the difference between these two and NMR. In NMR you are looking at a protein in the solution state, meaning the protein is in its native form, because in our cell the protein is always along with water. So, we are looking at in a native form in NMR, in solution NMR spectroscopy. So, therefore, we are looking at the native form of the protein, and the complete dynamics of the protein is also available to us, whereas in these two techniques the molecules are frozen. So, in that sense we are getting a snapshot, not a dynamics. We will not get the dynamics of the protein, we get a snapshot of the protein structure. So, that is the difference, whereas NMR helps us a lot. But again, as I said, NMR has its own limitations. So, one has to use this in a complementary manner. Now, one can use a purely computational approach. You do not have to actually do any experiment, do not have to make any sample, you do not need all these instruments. You can simply sit on a computer and get the structure of a protein, again in very high resolution, and that is using a homology modeling concept. Homology modeling means you have another protein which has a similar looking, which is similar identical sequence. Remember, we are looking at primary sequence, we do not have the structure, but if the primary sequence is very similar or identical to another protein for which the structure is known. So, let us take a protein A and protein B. If protein A has sequence matches with protein B to some extent, and what is that extent? At least more than 30% identity, means at 30% locations or more the protein A and protein B have identical amino acid. So, such a case, if protein B structure is known, then protein A structure can be solved by simply going to a computer. You do not need to really take protein A and go through all these experiments. But of course, the real reliability of homology modeling goes down if the identity is less than 30%. So, if it is 90% or 95% identity, two protein A and B are 95% let us say identical, means only in 5% of their residues are different, then most likely they will always have the same structure. So, that can be now determined with just a computer. That saves lots of time, and that also increases the speed, because this is faster compared to these techniques. Now, there is another option we can do, which has again come up in recent days, is that you take homology modeling, which is already taking the known structures and sequence, but you combine with some NMR data. Let us say you have some preliminary NMR data of that protein A and B. Suppose we are let us say we are looking at protein A and B, and protein A is known structure, okay, and protein B is unknown, and protein B as having a very high sequence identity with protein A. So, homology modeling can be done like here, but additionally I can take some more information by recording data on protein B. Okay. So, if I record NMR data of protein B and get some proton carbon experiments and spectra and chemical shifts, I can use that information and further improve my modeling. So, that helps to improve the modeling, and that is a combination of these two, homology modeling and NMR data. So, this is like is a combination of experimental and theoretical. So, it is not purely a theoretical approach, it uses the chemical shift knowledge which comes from experimental data. So, these are broadly the different techniques which are nowadays available to solve protein structures. But actually what do you do with this protein structure? So, once you have a protein structure, what do you do with that? So, typically these have to be deposited in a data bank. Okay. So, that is the most popular data bank is called the protein data bank. In a protein data bank, the protein structures are deposited, and you can see as of now, that is just recently, there are about 150,000, nearly 150,000 structures in this data bank. So, I would recommend very strongly to you to visit this website, which is this shown here, and actually have a look at this, what are the different types of information structures available, because this is where the history of data of protein structures are available, and this is where you have to deposit all the protein structures before you publish. So, if you have a new protein and you have solved it by NMR or crystallography or so on, you need to deposit here and get it verified whether that protein structure is correct or not by different techniques, and then only you can go ahead and publish. So, in the next class we will continue with the basic protein structure and we will see what are the different types of proteins. There is another class which we have not looked at, we will look at that and then continue with how NMR can be used for protein structure determination.